Hmm?
Hello, good evening, students. Today we are going to continue with our last class where we have left. We have started to discuss about hormone and the drugs related to hormone. So in previous two classes, I have explained you what are hormones, what are the sites of hormone of secretion, where that hormone is going to work, that means the active site, how that hormone are going to show its action by different mechanism of action, that also I have discussed. Then I have discussed about different hormones excreted from different endocrine system, and also I have explained about their individual work and functional and importance in our body. Today we will continue with the next topic in that same chapter, that is hormone related drug. In hormone related drug, we are generally going to study about different drugs which are going to act same as the hormone in our body, or otherwise some drugs we will also study that will do the different work as of the hormones. So they are called hormone related drugs. In hormone related drugs, today what we are going to study, we are going to study today insulin and glucagon. We will study today about the drug related to that two hormone, that is insulin and glucagon. In previous class only I have told you that insulin and glucagon, they are the hormones secreted from the pancreas, and pancreas is a mixed gland where exocrine and endocrine both properties are there. In endocrine, islets of Langerhans are there. In islets of Langerhans, alpha cell, beta cell, gamma, delta all cells are there. From alpha it secrete glucagon, from beta it secrete insulin. So today we are going to discuss about the action of insulin and glucagon and the drugs related to insulin and glucagon. So first of all we have to know that why we have to use that insulin, or why our body required insulin. In our body, a very important part is glucose. So for our brain, so for our head and for our nervous system, glucose is a very important part, and our head continuous need the supply of glucose. So our body have to maintain the glucose level into a certain amount, because anything is not very good above a beyond limit. In our body glucose is very important, but it is also have a limit on which that glucose react in our body better. More than that limit, it can cause different effect or different disorder or different irregular activities or undesirable or undesirable action in our body. So we have to maintain the glucose. What are the normal range of glucose? That is 90 to 120 mg, or you can tell 80 to 1 mg, 120 mg per ml of blood. That is the limit. If that blood glucose level increase above the 120 mg per ml blood, then it can call that in blood sugar level increases, that called increased blood sugar level, and in scientific term or medical term we call as diabetes, and another name it is called as hyperglycemia. So from the name only you can understand, hyper means more, glycemic means blood glucose. If the blood glucose level in our body increases than the normal limit, which is 80 to 120 mg per ml, then it causes the more sugar level in blood, that is very toxic for our body, and that condition is called as diabetes or hyperglycemia. So diabetes, it is a condition where our body blood glucose level increases more than its limit, which is 80 to 120 mg per ml. So diabetes mellitus is the disease where our blood level increases more than 90 to 120 mg per ml limit. It crosses the blood sugar level. When the blood sugar level is more than 120 mg per ml, it called a condition is diabetes mellitus or hyperglycemia. The diabetes mellitus are also two types. One is diabetes 1 and diabetes 2. The diabetes 1 is called as insulin dependent diabetes mellitus, and second one is called insulin independent diabetes mellitus. What is the difference between two? In diabetes mellitus 1, it generally occurs due to the lack of insulin. And we all know in blood generally glucagon increases the blood glucose level and insulin decreases the blood glucose level. So whenever insulin is not there in our body, that time it can cause increase in blood glucose level, that is called diabetes mellitus. That is the first type of diabetes mellitus or DM1 or insulin dependent diabetes mellitus. And second type is insulin independent. In that means in here our body have sufficient amount of insulin, but our body can't utilize or can't use that insulin. In this case, insulin can't do increasement of in our body and can't lead to the decrease in blood glucose level. So it can cause leads to the DM2 or diabetes mellitus 2 or insulin independent diabetes mellitus. I have told you that in our body insulin and glucagon, they control the blood sugar level, one by increasing blood sugar, another one is by decreasing blood sugar. So how that will work? In our body generally two types are there, both are from pancreas, Langerhans islands. From alpha we can get glucagon, from beta we got insulin. Now what this glucagon will do? That glucagon will increase the blood sugar level. And what insulin will do? Insulin will do the opposite work. It will decrease the blood sugar level. So whenever in our body glucose is less, it activates the alpha cell, and activation of alpha cell leads to the secretion of glucagon, and it will leads to the secretion of blood sugar level. And if our body have less number of glucose or less number of blood sugar, then it will stimulate glucagon, and if that glucose is more in amount, that will leads to the activation of beta cell, and it will increase the secretion of insulin, and secretion of insulin will leads to the less blood sugar synthesis. That means in our blood sugar will be less. So that is how glucagon and insulin maintain the bloody glucose. So I have told you insulin generally decreases the blood sugar level. So first of all we have to understand what is insulin and how insulin generally release. So insulin generally released from the beta cell of islets of Langerhans in pancreas. Now how that beta cell will secrete insulin, that we will study now with a simple diagram. Now we'll draw a simple diagram. Just suppose that is a beta cell. That is a beta cell. That is a beta cell. What is that? I will tell you later when I will describe about the diagram. So that is a beta cell, and this is beta cell membrane. Now in this beta cell membrane, that is the way in which generally glucose passes. In general glucose passes. That is the calcium channel ion gate. That is potassium ion channel gate. So I've told you in previous slide only, when that insulin will work, when the blood sugar level is increased, when that blood sugar level is increased, due to, it can be due to the diet, or it can be due to the overeating. So which reason, it doesn't matter. If the sugar is increases, it will leads to the activation of beta cell. And activation of beta cell, what it will do, that increased glucose will enters to this way. That glucose will leads to glycolysis, then it will leads to the TCA cycle and oxidative phosphorylation. That oxidative phosphorylation will increase the ATP-ADP ratio. Now, due to the increase in ATP-ADP ratio, what it will do? It will close the K plus gate. If it close the K plus gate, that means the K plus in the cell can't go pass through that gate. If that K plus is can't get out from here, then the K plus concentration in the beta cell will increase. If that K plus is increasing, that will leads to the depolarization, and that depolarization, what it will do? That depolarization will leads to the opening of calcium ion gate. If that calcium ion channel gate is opened by that depolarization caused by the over accumulation of K plus into the beta cell, it will leads to the influx of calcium. So it will cause the entry of calcium. That increased calcium will trigger some granules present here. That beta cell granules is generally connected to the beta cell membrane, and that granules generally contains insulin. When that Ca2 plus concentration is increasing and K plus also is increasing into the beta cell, it will lead to the secretion of insulin. That secretion of insulin will lead to the decrease in blood glucose level. So this is how insulin is generally released from the beta cell. So these were the simple diagram by which I have explained you what is the mechanism of insulin release from the beta cell. Now, when the blood glucose level is increases more than that its limit, that means 80 to 120 mg per ml, it is a condition of diabetes mellitus. So to treat diabetes mellitus, there are different types of drugs that are available into the markets, and all the drugs are classified into some classes. Now we'll discuss about the classes. First class is that is generally used to treat diabetes mellitus, that is sulfonylurea. In sulfonylurea, two types are there. First generation are there, second generation are there. The example of sulfonylurea is tolbutamide, and the second gen example is glipizide. That is the first classification of anti-diabetic drugs used to treat diabetes mellitus. The second type of classification in anti-diabetic drugs are meglitinides. In meglitinides, examples are repaglinides, nateglinides. So that were the second types of anti-diabetic drugs, that is meglitinides. Example are repaglinides and nateglinides. That is the second type of anti-diabetic drugs that is used to treat diabetes or hyperglycemia. Then the third type of anti-diabetic example is, or classification is, biguanides. What are the example of biguanides anti-diabetic drugs? Example are metformin and penformin. Metformin is mostly used as a biguanide in case of diabetes mellitus to treat diabetes or hyperglycemia. Fourth type of anti-diabetic drugs are diazolidine-diones. So that are the fourth type of anti-diabetic drugs, that is diazolidine-diones. In that, examples are rosiglitazone, pioglitazone. So the fourth type of anti-diabetic drugs classification is diazolidine-diones. Examples are rosiglitazone and pioglitazone. That rosiglitazone and pioglitazone, they are generally used as diazolidine-diones anti-diabetic drugs to treat diabetes or hyperglycemia. The fourth type of drugs are, or fifth type of drugs are, alpha glucooxidase inhibitors. So what are the example of alpha glucooxidase inhibitors? Acrabose, Vogelbose. So acrabose and Vogelbose is used to treat anti-diabetic drugs as a to treat diabetes as alpha glucooxidase inhibitor to treat diabetes mellitus or hyperglycemia. So what are the five types of anti-diabetic drugs we have studied here? Hyperglycemia, to treat that one, first one is sulfonylurea. Second one is meglitinides. Third one is biguanides. Fourth one is diazolidine-diones. And fifth type we have studied alpha glucooxidase inhibitors. So these are the five types of anti-diabetic drugs that mostly used to treat diabetes as a anti-diabetic drugs to treat diabetes and hyperglycemia. The most important and most used are sulfonylurea. Example I have told you, tolbutamide. Tolbutamide or sulfonylureas are the most used anti-diabetic drugs in the daily life. So now we will study about its mechanism of action. That mechanism of action is almost same as insulin release. That is a beta cell. When sulfonylurea is introduced into the beta cell, that will close the calcium channel gate. That means the calcium will not pass through that K plus channel. That means into the beta cell K plus will increase, ATP will increase, and it will leads to the depolarization, and lead to the depolarization that Ca2 plus gate will open. If that calcium channel gate is opening, that means the calcium channel from the outside, calcium will increase into the beta cell, increasing ATP, increasing calcium ion, and increasing K plus ion into the beta cell will leads to the release of insulin from the beta cell granules, and that insulin further will leads to the decrease in blood glucose level, and thus how sulfonylurea used to treat diabetes mellitus or hyperglycemia. So that is the simple mechanism of tolbutamide or other sulfonylureas. Now we will study about the pharmacokinetics of tolbutamide. The tolbutamide is generally absorbed in stomach. That generally absorbed in stomach. That distribution is almost 90 to 99% is distribution because of the protein bind, and due to that high protein bind the bioavailability is 90%, and due to that bioavailability the peak blood concentration to conquer it will take 4 to 6 hours. Then after distribution throughout the body it will show its action, and after its action it will get metabolized into the liver by CYP C29 enzyme, and after metabolism it will excreted in unchanged condition in small amount by urine by kidney. So these are the pharmacokinetics properties of tolbutamide: absorption in stomach, high bioavailability and protein binding, metabolism by the liver CYP29 enzyme, metabolized in liver and then excreted in small amount in unchanged form from the kidney through urine. So that is how tolbutamide generally works as sulfonylurea to treat diabetes. What are the adverse effects of tolbutamide? Nausea, vomiting, excess use can lead to the hypoglycemia, skin rashes. In old people it can cause ischemia, or it can cause also different cardiac problem. So these are the critical adverse effect in case of excess use of tolbutamide or any sulfonylurea drug. So by that I have finished off the sulfonylurea as a anti-diabetic drug. So that was the process by which how generally insulin and the sulfonylurea generally decreases the glucose level. Now we'll study about the glucagon and how it regulates the blood sugar level with insulin. I have told you insulin generally decreases the blood sugar. Now if the blood sugar level is very decreased below the 80 mg per ml, that will cause to hypoglycemia. So to treat that time glucagon will increase in more amount from the activated alpha cell. So due to the over accumulation of glucagon that will binds to with G protein couple receptor, and binding with G protein couple receptor, that glucagon will helps to increases blood sugar level. To increase the blood sugar level, glucagon have different types of mechanism of action, how by which generally that glucagon increases the blood glucose level. First one is it promotes gluconeogenesis. What is gluconeogenesis? We all know from known sugar source, that means from it can be amino acid or it can be protein or it can be lipid, it will produce or it will convert it to glucose, that is called gluconeogenesis. So glucagon will increases that process and it will leads to the over accumulation or over formation of glucose, and that will use to treat hypoglycemia. The second type of mechanism that generally glucagon do to increase the blood sugar level in case of hypoglycemia, that is by inhibiting glycolysis. We all know in glycolysis what it will happen. In glycolysis, glucose generally breaks down into the pyruvate or lactate. That means that glucose will decreases. What glucose will do? That glucose will decreases the glycolysis, and the inhibition of glycolysis will lead to the decrease in glucose to pyruvate lactate conversion, and it will lead to the accumulation of glucose, and that will treat hypoglycemia. The third type of mechanism by which that glucagon will control or will increase the blood glucose level, that is by inhibiting glycogenolysis. By promoting glycogenolysis. In that glycogenolysis, generally glycogen will convert it to glucose. So glucagon will increase the gluconeogenesis and it will conversion from glycogen to glucose. That glucose will increase the blood sugar level and it will treat hypoglycemia. The fourth type of mechanism that generally glucagon also use, that is inhibiting glycogenesis. In that, what generally happens? Glucose generally converted to glycogen. So what glucagon will do? Glucagon will decreases the glycogenesis process and it will leads to the non-conversion of glucose to glycogen. That means glucose level in the blood will increases and blood sugar level also will increases and it will treat hypoglycemia. So these are the mechanism of action of glucagon by which it generally increase the blood sugar level. First one is by promoting glucogenolysis, second one is by promoting glycogenesis, third one is by inhibiting glycolysis, and fourth one is by inhibiting glycogenolysis. So that are the four processes by which generally glucagon increases the blood sugar level. And by that today's video I have explained you about how insulin and glucagon in our body is controlling blood sugar level and maintaining blood sugar level in our body to 80 to 120 mg per ml by their collective effort. And also we have studied about different drugs which are generally used when the blood sugar level is increased in our body. That condition is called diabetes mellitus. About that drugs and their classification, about their mechanism of action, and about insulin and insulin mechanism of action and release mechanism, all we have studied in today's video. So in next class I will continue with the next continuation, that is drugs is related to hormone, that is thyroid hormone or thyroxine. That I will continue in the next class about the drug related to thyroxine hormone. Thank you.